Hello dear students, welcome back to another learning lessons in our Physio-Pulse channels. Today we will discuss about very interesting topics, that is lipoproteins, in a simple, easy to understand way for your exam purpose. Moreover, you will find the clear explanations, diagram and important concepts during these lectures. Before we begin, if you find this video helpful, please like, share and subscribe, support the channel. And now let us understand about the lipoprotein. Actually, lipoproteins are the spherical macromolecules complex which is combined with the lipids and proteins. So apolipoprotein and apoproteins both are combined here, so it is called the lipoprotein. There are four main types of lipoproteins, and before that, if you want to see the apo enzymes, that is the ApoA, ApoB, ApoC, ApoD, and ApoE, all are in here in this lipoprotein structures. So there are the main major four types of lipoproteins. There is the chylomicrons one, then VLDL, very low density lipoproteins, LDL, low density lipoprotein, and HDL, that is the high density lipoproteins. So one by one we will discuss about here. So lipids are absorbed from the diet. When we eat something, the lipids are absorbed, and also lipids can synthesize in our liver, adipose tissue. So there are the two ways. There is one is the exogenous pathway, another is the endogenous pathway. So they are transported between the various cells and organs for the utilization and storage. Lipids are insoluble in water, and the problem of transportation in aqueous plasma is solved by associating nonpolar lipids, that is called the triglycerides or triglycerols, and cholesterol esters. And with their amphipathic lipids, that is phospholipid cholesterol, and proteins to make what is miscible in lipoproteins. So these are lipoproteins how we get from the diet as a source, and another thing, it can be synthesized in the liver, and it can be transported between the various cells, organs, and utilization of the and storage. Now, general structure of lipoprotein, if you see, then the lipoprotein consists of a nonpolar core groups and a single surface of amphipathic lipids. The nonpolar lipid cores consist mainly triglycerol or triglycerides, and which comes from the monoacylglycerols, then diacylglycerols, and that the synthesis of the triacylglycerol, and cholesterol esters also, which is surrounded by a single surface layer, amphipathic, that is phospholipid and cholesterol molecules. These are oriented so that their polar groups face outwards of an aqueous medium, and the protein moiety of the lipoprotein is known as the apolipoprotein or apoprotein. So this is the general structure of lipoproteins, and here the triglycerides, these are the triglycerols, these are proteins, they are embedded here, and phospholipids like core groups, free cholesterol is moving here, and cholesterol bound with the fatty acid or fatty acids. So this is the general structure of the lipoprotein. Now, if you see the classification of lipoproteins, what we see, lipoproteins are differing in the lipid and protein density, size, and site of origins. So lipoproteins may be classified as follows, listed from the larger and less dense to smaller and denser. So lipoproteins are larger and less dense when fat to the protein ratio is increased. First of all, first classification is the chylomicron. These are the largest size and have low density. So large amount of the lipid, they carry the triglycerides, dietary fat, from the intestine to the liver and the adipose tissue. Second is the VLDL. Already we know the full form of VLDL, very low density lipoproteins. They carry the triglycerides from the liver to the adipose tissue. And intermediate or IDL is derived from the catabolism of VLDL with a density of ranging intermediate between the VLDL, that is very low density lipoprotein, and low density lipoprotein. So IDL is the in between LDL and VLDL. They are not usually detectable in the blood. So number 4 is the low density lipoprotein, which carry the cholesterol from the liver to the cell of the body, and the LDL is sometimes referred to as the bad cholesterol. So this is the very bad, because this cholesterol comes from the liver to deposit into the adipose tissue of the cells. So therefore, it can happen here. It will lead to progress the atherosclerosis. In case of HDL or high density lipoproteins, these are the smallest in size and has high density, large amount of protein. They collect the cholesterols from the body's tissues and bring back to the liver. So during the deposition of cholesterol into the arterial wall or the any adipose tissue, it can bring back to the liver. So this type of cholesterol is very good cholesterol in our body. So this is called the friendly cholesterol or healthy cholesterol. Now these are the classification. The classification which are as per the density level. Higher is the chylomicron, lower is the HDL cholesterol. Now, lipid profile. What is the meaning of lipid profile? It consists of the cholesterol, triglycerols, and as well as the some LDL or HDL cholesterol. So these can how much amount present into the body or blood that can be determined through these lipid profiles. So whenever in our body there, if you see the higher, if you see the abnormal concentration of the lipids like the cholesterol, triglycerides, then it will lead to the some problems like the cardiac problem, atherosclerosis, etc. It can lead. So increase and decrease level usually occurs abnormalities of synthesis, mainly synthesis, degradation, and transport of these substances like the lipoprotein particles. So according to this, it can be called or it can be named as the hyperlipoproteinemia. That means in the blood, lipoprotein is very much higher than the normal level, and hypolipoproteinemia, that is below the level of normal, that is the lipoprotein level is normal in below the level in blood. So lipid profile measures the total amount of cholesterol in the blood, number one. The level of HDL cholesterol, how much amount, that is the HDL cholesterol or good cholesterol. The level of LDL cholesterol, that is the bad cholesterol, and the amount of triglycerides or fat present. So these are the four important markers by which we can measure, and after measuring the data, we can say the lipid profile is good or bad. Now, HDL, high density lipoprotein, that is the HDL is one of the four major groups of lipoproteins. HDL is composed of the 50% protein, 25% phospholipids, 20% cholesterol, and 5% triglycerides. So HDL, good cholesterol, carry the cholesterol from the body cells to the liver, where it catabolizes and excreted into the bile. It removes the excess cholesterol from the tissue, so it cleans the blood. High level of HDL is better because it reduces the risk of heart and the blood vessels disease. Now, how to measure this? This is measured through the process sample collections, blood, lipid profile involves measurement of total cholesterol, triglyceride. These after 12 hours fasting. And how to estimate this? This is through the ultracentrifugation, electrophoresis, or chemical precipitations. These are the methods by which we can determine the concentration of the LDL, HDL, triglyceride, cholesterol, etc. So in this way, we can calculate HDL, LDL, VLDL, even also triglyceride, total cholesterol. If you see the LDL, what is the meaning of LDL? Already we told that it is a bad cholesterol and carry the cholesterol from the liver to the blood. So blood to the organ. So it has less protein content and more content of cholesterol. LDL cholesterol is easy to stick in the wall of blood vessels. So reducing level of LDL cholesterol is the major treatment for targeted the cholesterol lowering medications. So we are taking the medications to reduce the LDL cholesterol. Like the statin, we have used atorvastatin, rosuvastatin, all the statins we used to reduce the LDL cholesterols. And this is the normal ranges. HDL milligram per deciliter. Desirable HDL is greater than 60. Low HDL is less than 40. And desirable LDL is less than 110. And high LDL is greater than 130. Concentration of LDL, it can be calculated this way. These equations, LDL is equal to triglyceride minus HDL plus VLDL. That we can. And so, students, we already covered all the lipoproteins, all the lipoproteins like the LDL, HDL, VLDL, triglycerides, and total cholesterols, and the some desirable amounts. What we have seen that in case of HDL cholesterol or in case of LDL cholesterol. So I think all this important data in your exam purpose, it is also very important. So if you found these lectures helpful, do not forget to like, share, and subscribe to the channel. Press the notification bell so you never miss our upcoming biology, physiology, or biochemical lectures. So thank you for watching, and we'll see you in our next class. Thank you.